some announcements first. So I guess you can see my screen now. If you haven't followed us on social media, you can follow us on Facebook, Twitter, LinkedIn, and on Instagram. And also, if you want, you can watch some of our previous events on our YouTube channel. Also, I would like to recommend you to follow us on Instagram, because we are now having a new live event series on Instagram. So especially this week, we are gonna have on Sunday a new live event with one of our previous members, Tülay Karakulak. She's gonna tell us about her experience as a PhD student, as a bioinformatician. And I think it's going to be great. And also, I would like to remind that it's going to be in Turkish. So we are sorry for our international followers. Also, I would like you to recommend you to follow us and visit rsgturkey.com. And in rsg-Turkey.com, you can find valuable information on bioinformatics. Also, you can become a member of our family. You can be either a passive member and hear about us more, or you can be an active member and organize these nice events with us. So like usual, we're going to have your questions at the end of the session. So you can use the chat box to ask your questions. And if you use this little hand icon down below before writing your questions, we will know that somebody is asking questions. So we are going to wait you until you write your question. So as I mentioned before, we have Dr. Merve Kılınç with us today. Gülşah Merve Kılınç got her bachelor's degree from Hacettepe University Department of Biology. Then she got her PhD in molecular biology and genetics from Bilkent University, and then she worked as a postdoctoral researcher in Middle East Technical University, and then she continued her postdoctoral studies at Stockholm University at Department of Archaeology and Classical Studies, and now she is in Hacettepe University. Dr. Gülşah Merve Kılınç, I would like to thank you on behalf of RSG-Türkiye for taking that time and making this webinar. I don't want to steal your presentation time, so stage is yours.
Thank you very much. I also would like to thank you and to ISCB RSG-Türkiye for this kind invitation. And also thanks everyone for joining us today. Now I can share my screen. I will try it, maybe entire screen. You still don't see my screen, right?
I guess right now we can see it. No, it's gone.
Now? Now do you see the screen? Or?
Yes, now we can see it.
Okay. So thanks again for your kind invitation. And today I will be telling you a story about the ancient human populations from Northeast Asia, especially from Siberia, who lived during the Holocene time period. And the topic will be covering some ancient genomics methodologies. Because of that, I first would like to give you a very brief background information about what is ancient genomics and what is ancient DNA and what kind of questions do we ask in this field and what is our approach to find the result. So basically it's a bit a recent field, but not that much recent, which is very popular over the last 2 decades maybe. And the ancient genomics allows us to read our past at some level. So we can travel back in time, we can go back to the Middle Ages, Iron Age, Bronze Age, Neolithic period, even as back to the late Upper Paleolithic and Mesolithic time periods, and we can analyze, we can examine the DNA of the past populations, both the humans and animals and plants. We can analyze their DNA. So at this point, the ancient DNA is, you can think it as a kind of time capsule that allows us to do this travel back in time. So this is a regular DNA, but this time the source of the DNA is archaeological or historical materials. So you can get this kind of ancient DNA from museum specimens or zooarchaeological material, archaeological findings. You can obtain ancient DNA also from the fossil remains, mummified tissues, coprolites, and sediments. So those sources can provide ancient DNA to us. And what we do is to produce the DNA sequences, and our aim is to prevent the modern DNA contamination and to produce authentic DNA sequences when we are doing the ancient genomics studies, because we just we need to get the DNA sequence that is exactly coming from the old individuals. So we need to prevent the modern contamination. To do that, there are some kind of things that we can do. The first thing that we do is excavating the samples and curating them under clean conditions. And also, after excavating and curating them, when we bring them to the lab, this should be a kind of physically isolated, clean lab facility that you don't have any other modern contaminants. And the surfaces, tools that we use, and also the samples are decontaminated using some techniques. Even under these conditions, even under the precautions that we take to prevent the modern DNA contamination, there is always at some level of, there can be, not always, but there can be some level of contamination. At this point, we need to detect it and quantify the level of it. So for that one, we do some kind of experimental approaches in our isolation. And we need to also show that the results that we obtained are consistent with the patterns of ancient DNA. So we need to quantify the template DNA. For that, generally the bioanalyzer are used. And also we can try to observe the age-dependent patterns that we see in the DNA. Here the aim is, after doing all these steps and isolating the DNA, what we do in ancient genomic studies is to sequence the ancient DNA, which means that we try to find the complete sequence of A, C, G, and T's in the DNA using a next generation sequencing platform. While doing this, after when we sequence the DNA, the first question that comes to our mind is that we need to consider is the question of, is the DNA sequence that we produced here is really authentic? Which means that is the sequence coming from an ancient organism, or is it coming from a modern human, for example, running the experiments in the lab? First, I would like to show you the figure B here. This is from the Human Evolutionary Genetics book. It shows that when you sequence the ancient DNA, this is the sequencing of ancient DNA, and from Homo sapiens, and there is the reads, sequencing reads that are coming from the Homo sapiensis here, but there you see some kind of contamination, and the sources are like bacteria or other eukaryote, or some sequences that we cannot assign. And even in the bacterial part of the DNA sequences, you can see that there are different kind of bacteria DNA sequences. So this kind of contamination that is coming from bacteria is not a big problem if you are working with ancient human genomes, because you are trying to find the human population dynamics. So these sequences can be ignored. But the other problem that is more important than this is a modern human contamination. This figure is from Skoglund et al. From PNAS 2014, and it shows that, for example, here if we examine the pink or purple points. When there isn't any contamination, when the contamination level is 0%, then that individual, this is a PCA that shows the individual the distributions of the individuals depending on their clustering of the individuals depending on their genetic makeup. So this individual, when there isn't any contamination, it seems like the individual is here on the PCA. So you can say that it has affinity to kind of the northern populations, for example. And then in the paper, when they artificially introduce the contaminating sequences to the individual, like 20% contamination, 50% contamination, and 90% contamination from an individual from France, then genetic affinity of this individual is coming closer to the French populations. So this kind of contamination that is coming from modern humans can interfere with the interpretation of our population genetics analysis results. And to understand, so to prevent this, we take those precautions that I showed you in the previous slide. But even under these precautions, even if there is contamination, what we do is to examine some ancient DNA specific damage patterns. At this point, one of the most important signs that we need to check, first one is that the distribution of the read lengths. This figure is showing the distribution of the sequencing reads that are coming from an ancient individual. The x-axis is showing the length of the reads starting from 50 base pair to 300 base pair, and the y-axis is showing the number of reads. So we expect some kind of this kind of distribution, like the mean read length is changing between 50 or 60 nucleotides, because the ancient DNA is degraded. It is found as like smaller fragments. So we need to see this pattern in ancient DNA sequences to be sure about its authenticity. This is one of the signs. And the other sign of the damage that is specific to ancient DNA is that increase in the C to T type of transitions at the 5' ends of the nucleotides. Here, this shows, the x-axis shows you the distance from the 5' end of the sequencing read, and the y-axis is showing the frequency of the nucleotides C, G, A, and T. When there is C at the reference sequence. So we see that there is an increase in the C to T type transitions at 5' ends because of the post-mortem damage of the DNA. So these two signs can be like two first initial points that we need to check to be sure about if we sequenced really authentic DNA or if there is some contamination. There are also some other methods that we can run to understand if the DNA is authentic based on the mitochondrial DNA or nuclear DNA. So depending on that, we decide if the DNA is authentic. And after being sure that there isn't any contamination, what we do in these studies is to discover the genomic variants. So normally, if you sequence an individual's genome who is living today, the thing that you do is sequence the genome, map it to the reference genome, and then call the SNPs and indels in the genome. And to do that, you can use some softwares like GATK or like SAM tools. But in the case of ancient DNA, we do some more things to be sure about the SNPs, because the ancient DNA is degraded and there can be also contamination. So what we do is to use a kind of reference panel to reduce the false positives. So for example, there is an array of human origins array, which includes SNP genotype data for 2,000 individuals for more than 500 SNPs. So what we do is trying to find the genetic variants in our ancient samples. In case of humans, I'm talking for humans. So if you sequence an ancient human genome, what we do is finding its SNPs that are overlapping with this SNP data set to be sure that we are really working with the SNPs that are already known in the present day populations. Or there are some other panels like 1000 Genomes Project, which has whole genome sequence data. And after finding the SNPs of the individuals that are overlapping with the reference data sets, we prepare large genomic data sets that includes both the ancient individuals that we sequenced in our study, and also we merge them with published ancient individuals from all around the world to prepare a comprehensive data set to make some comparative analysis. And also we merge them with the modern individuals. Based on these approaches, we can ask so many different questions about the dynamics of the past human populations. And so far, I have worked with multiple ancient human genomes from different parts of the world, including Anatolia, Eurasia, Scandinavia, and Siberia. Today, the story from the ancient genomes that we learned, I will tell you, is about ancient Siberia. Siberia is a very large geographical area spanning this part of the world, and the region has been home to very different, multiple ethnic groups, so many different human populations throughout the prehistory. So the region has a very dynamic population structure, starting from the late Upper Palaeolithic, coming to the present days. The region is important because we know that there is the earliest traces of the post-last glacial maximum human occupation, and also you can find the ancestors of the Palaeo-Inuits in this region too. And the population history throughout the region is characterized by multiple gene flow and admixture events. And today, to understand the population history, I will tell you the story of genomes spanning a time period between late Upper Paleolithic and Middle Ages in Siberia. So this is a published study in 2000, very early this year in Science Advances. In that one, what we do is first focusing on 3 different geographical regions in Siberia. The first one is the orange. So each point represents one single ancient individual, and here we have the timeline that is showing the ages of the individuals. The orange color is here showing the individuals that are coming from the Yakutia region, which is here, like this gray shades. This region is known as Yakutia. So we have individuals from here, and also we have some here. You have the Lake Baikal, which is a very famous lake and the deepest lake of Earth. We have 2 different populations from around the Lake Baikal. The populations that are coming from the west of the lake are named as Cispecal populations, and the populations that are coming from the east of the Lake Baikal are called as Transbaikal populations. To investigate the population dynamics in Holocene Northeast Asia, what we did is to sequence the genomes of all these 40 individuals up to 15x coverage. So the coverages ranged starting from 0.03 to 15x. The maximum coverage for the Yakutia was almost 9x. For Transbaikal, it was like almost 5x coverage, and we generated data almost 15x for the Cispecal populations. So during the talk, I will first tell you about the population dynamics of the Yakutia, and then I will proceed to the Baikal population. So when we check the Yakutia populations, the story starts here in a cave, which is known as a Kayırgas cave, and the name of the individual here that we gave this name to that individual is Kayırgas 1, and this individual is almost 17,000 years old. And then we have some other subsequent human populations spanning a time period between almost 7,000 years ago and 3,000 years ago. We also have one Iron Age individual from 2,500 years ago. And we investigated the population transformations for these Yakutia populations. So this Kayırgas 1 individual is is coming from a place, a geographical area, which is one of the first known post-last glacial maximum representatives of the settlers of the central Siberian plateau. The first thing what we did is to run a principal component analysis. Here we see the results for the principal component analysis. Each point represents one individual, ancient or modern. And here we have the carrier gas one individual. So this is summarizing the genetic affinities between the ancient and modern populations. In that case, what we see is that the carrier gas one individual is located sitting on the PCA between two distinct lineages that are coming from the same region. Maybe I can go back one slide back. So here we have Yana Upper Paleolithic, and here we have the Kolyma individual from Mesolithic period. And here we have our carrier gas one individual. And when we check the PCA, we see that carrier gas one is sitting between the Yana, which is representing the ancient North Siberians, who are the first inhabitants of the Siberia, represented by 38,000-year-old individuals here. And then between Yana and Kolyma. And Kolyma is representing the ancestors of the Native Americans, which is called as a lineage as ancient Paleo-Siberians. So we can say that the carrier gas one here in this region, in this large geographical area, is marking the first genetic shift here, because we have 38,000 years ago, the genetic structure was like here, and then there is a shift from Yana, from Yana to carrier gas, and from carrier gas to Kolyma one. And when we run the F3 statistic, which measures the shared alleles between populations, we observe that this individual shared more alleles with the Native American populations, which is also located here on the PCA. We also observe that when we run the F4 statistics to measure the shared genetic drift between the carrier gas one individual and the Kolyma individual here, like the ancestors of Native Americans, we observe that carrier gas one and Kolyma are forming a clade compared to the other populations, other late Upper Paleolithic populations from West Eurasia. And also this individual here shared more alleles with the ancient North Eurasians from Eurasia compared to the first inhabitants of the region. So we, based on this pattern, we also wondered about where the individual will be fitted on an admixture graph, and we fitted a tree using a tree mix, and we observed that the carrier gas one is forming a distinct lineage compared to the Kolyma population, like the ancestors of Native Americans. And also what we observed is that this new lineage is contributing to the later coming populations from represented by Kolyma individual, which we observed using the QPADM analysis. In QPADM, what we observed is that we modeled the Kolyma individual, like the native ancestors of the Native Americans, as a mixture of this newly reported carrier gas one individual and the East Asian populations, which are represented by the Devil's Cave populations, which is here. And after that, what we wondered was the population transformations in Yakutia, because we have one population here represented by Yana, and then other population from Siberia here, which is represented by Kolyma individual. We have our carrier gas one individual. Then we produce genomic sequences for the later coming populations, which are spanning 17,000 years and also 4,000 years before present. So you can also remember them from the map. Here we have the carrier gas one and then the later coming populations in this region. So if we come back here, and what we see is that genetically the later coming populations of Yakutia were distinct from the first inhabitants of the region from carrier gas one. And also archaeologically, what we know is that between 13,000 years and 2,500 years ago, there has been a period of like significant cultural and social changes during the transition from Palaeolithic to the Neolithic. And this archaeological data here is supported by the genetic data because we see that they are genetically also very different. So there have been three different major shifts in the Yakutia populations. The first one is that from Kyrgyzstan, from Yana to Kyrgyzstan, from Kyrgyz to Kolyma, there is another shift. And when we see that around 7,000 years ago, the populations are becoming a bit different here. Then around 4,000 years ago, populations are becoming more and more different compared to the previous populations. And these are the results of the admixture analysis that shows the genetic components in the genomes of the ancient individuals. Here, the pattern that we see is that, so this is the modern populations admixture analysis results for 14 ancestral clusters. And purple color here is representing an ancestral component that is coming from the Native American populations. And the yellow color here is representing the ancestral component that is representing the East Asian populations. And what we see here is that in time, starting from 17,000, when we come to 2,500 years ago, the amount of the East Asian component, the yellow component, is increasing in time, and the amount of the Native American component, which is the purple component, is decreasing in time. So we see that there is a change in the genetic structure of these populations. The other interesting question was about the Bacteriolithic complex. This is important because there has been several arguments implying that these individuals, human populations that are related to Bacteriolithic complex might be ancestors of Palaeo-Inuits who inhabit the Greenland. But there has not been genetic evidence for this archaeological claim until this time. And when we sequence the genomes of two individuals who are coming from a Bacteriolithic complex, we observe that these individuals had genetic affinities towards the Palaeo-Inuit populations, which is represented by the Sakak individual, which is a published individual. So this was the first indication of the Bacteriolithic complex can be genetically the ancestors of the Palaeo-Inuits. But to be sure about these results, we also run some formal tests of formal tests using F4 statistics, and we observed that the Belkachi individuals shared more genetic drift with the Sakak individual. And when we modeled the genome of Sakak individual using QPADM analysis as a mixture of Belkachi and West Eurasians, this is another individual, ancient individual that represents West Eurasian populations, we observed that the Palaeo-Inuits are a mixture of the Belkachi populations and the West Eurasians. So this was the first time that we were sure that the individuals who are related to Bacteriolithic complex are the genetically ancestors of the Palaeo-Inuits. And after resolving those transitions in Yakutia populations, we focused on the Baikal populations, which we investigated both the Cispecal, west of the Lake Baikal, and Transbaikal, the east of the Lake Baikal populations. Here we also have a very large geographical area and also very large time span because the individuals are starting from almost 8,500 years ago and coming to the Middle Ages like 600 years ago. Then we investigate the population transformations here, and the reason behind studying the Baikal populations is, first of all, we know that the Transbaikal area is a unique area on Earth to study the post-last glacial maximum human population dynamics because the first activities by humans after the last glacial maximum is indicated by the pre-Neolithic pottery of the individuals from here. And also we see the appearance of pre-Neolithic ceramics around 13,000 years ago in this Baikal area, which might be associated with possible migration from the Southeast Asia populations. So to this end, what we did is first examining the Transbaikal populations. Here we see that on PCA, they are clustering together, they are grouping together, forming a tight cluster. But there is one exception, which is a Transbaikal individual who is 8,000 years old. This is the oldest individual that we investigated from the Transbaikal area. And this individual appeared to be genetically more similar to the Yakutia individuals, the Palaeolithic complex associated individuals, and it is very distinct from the rest of the Transbaikal populations. So we can say that based on this result, we can say that the people who settled the Transbaikal area around 8,000 years ago can be ancestral to the other people who settled the Lena region those times. Even in the F4 statistics, when we measured the shared alleles between this Transbaikal individual and also the Balkachi individuals, we observed that this Transbaikal individual shared more alleles with these two Balkachi individuals. And after that, when we see this individual here, there is on the other side of the Transbaikal population, we see that there is a genetic continuity between 8.3 thousand and 3,000 years ago, and there isn't any major genetic shift in this Transbaikal area. So you can see that this individual is here, 8.5 thousand year old individual is seated here, but the rest of the Transbaikals are grouping here. So we can say that there is a genetic continuity amongst the Transbaikal populations. And also we observe that those populations, these Transbaikal individuals had genetic affinity towards the Devil's Cave individuals, who are the East Asian ancient individuals. And also, when we check the admixture components in their genomes, what we see is that, again, the increasing pattern of the East Asian component and decreasing pattern of the North Native American component. So we can say that, and also if you check this individual from this triangle individual from the map, you can see that this is from another region. Here this part is Transbaikal and this part is Amur Oblast region. On the map, it seems like they are a bit close, but in reality, they are very far away from each other. And we see that this Amur Oblast individual also had the same genetic affinity towards this Transbaikal group. So we can say that in this part of the Lake Baikal, in the east of the Lake Baikal, we see that there is a genetic continuity over a very long time period. Like starting from 8,000 years ago, coming to the 1,000 years ago, and also in a very large geographical area, we see that this genetic continuity, reaching from Transbaikal region to Far East. So this has a contrasting pattern compared to the Yakutia populations. When we also come back to the other side of the lake, so this was the east side of the lake and this is the west side of the lake, there is also again a contrasting pattern. So there has been multiple population transformations, and unlike the Transbaikal populations, this was also interesting to observe. So for the Cispecal individuals, what we did is producing genomic sequences for 20 individuals, spanning a time period between almost 9,000 years ago and 600 years ago, and the individuals are located here, this part of the world. And what we see here is that, unlike the Transbaikal populations, we see multiple population groups. If we examine it here, first we see that around 8,900 years ago, this Cispecal individual is grouping together with the Transbaikal individuals. So we can say that there has been a a uniform genetic pool around the Lake Baikal, or around both east and west sides of the Lake Baikal, almost 8,000 years ago, 8.9 thousand years ago. Then we see a genetic shift when we come to 7.9 thousand years ago, because the individual here, this is 7.9 thousand year old cyclodrome individual, it marks the first genetic shift here. From here, we are coming here, and this individual is grouping together with other individuals from Cispecal spanning between 7,000 and 6,000 years ago. And yet again, when we come back to the 6,000, 6.1 thousand and 3.7 thousand years ago, there is another group which is genetically closer to also the Palaeolithic complex individuals. So we see that three distinct groups spanning this time period. So based on time, they are grouping, and there is a shift in the genetic structure of the Cispecal population. So there is multiple population movements, but in contrast to the Transbaikal populations. And after that, what we did was to investigate the genetic diversity in all groups, like in Yakutia populations, Transbaikal and Cispecal populations. When we compare them, here we have one individual from European Neolithic populations, and here we have one individual from the European Mesolithic populations. Normally, we know that the European Mesolithic hunter-gatherers have low genetic diversity and low effective population size, and European Neolithic farmers has high genetic diversity compared to the Mesolithic individuals and high effective population size. Here the pattern is that for the Yakutia populations, Transbaikal and Cispecal populations, we see that they are sitting between those European farmers and European Mesolithic hunter-gatherer individuals. And the lowest diversity that we observed was in the Yakutia populations here, and also in the Transbaikal individual. For the rest of the populations, for the rest of the Cispecal populations, we see that they have higher levels of the heterozygosity, which means that those Cispecal populations were more diverse compared to the Transbaikal and Yakutia populations. This is not surprising because even on the PCA, we observe that there are multiple population transformations in Cispecal, so population movements are common. But in Transbaikal, we see that there is a compact one group of individuals, then the diversity can be, of course, lower compared to the Cispecal individuals. But there were two exceptions in the Cispecal region. Those were two individuals from Cispecal area, which are dated to 4.4 thousand years ago. Surprisingly, those individuals had low levels of heterozygosity compared to the Transbaikal individuals. And also we investigated the distribution of the long runs of homozygosity. Again, consistent with the heterozygosity estimates, we see that the Yakutia populations and the Transbaikal populations had more long runs of homozygosity, which means that they were more homogeneous populations compared to the Cispecal individuals. Again, when we check the effective population size changes over time using the MSMC analysis, what we observe here is that the Yakutia populations had lower effective population sizes compared to the Cispecal populations. For Transbaikal population, we couldn't run this analysis because of the coverage issues, so we picked the best coverage samples to run the MSMC analysis here. Here, since these two individuals were exceptions, which means that around 4,000 years ago in the Cispecal region, population size might be a bit lower compared to the other times of the Cispecal region. And what could be the reason? And then what we did was to investigate the possible presence of sequencing reads that are coming from a kind of microbe to the region. And what we did was we discovered that the Cispecal individual, one of them, which is dated to 4.4 thousand years ago, had reads related to Yersinia pestis. And also we discovered presence of Yersinia pestis in another individual that is coming from Yakutia. So this was like a speculation about was there a pandemic during the Bronze Age, in Cispecal and Yakutia regions, which resulted in the reduction of the effective population size, reduction in the size of the population. But this is just a speculation because we just discovered those reads in genomes of these two individuals, and we need to produce more data to be sure about the presence of this microbe during this time period. So as a result, what we see is that we investigated 3 different populations from Siberia and see a very different patterns of the population dynamics, like in Yakutia and in Cispecal, multiple population transformations and gene flow events. But in the Transbaikal region, more compact and low population we see there. So this was my, these were the results that I was going to tell you. So please, you can ask your questions if you have any. Thank you very much. I don't know, was it fast or unfortunately?
I guess you can hear me right now.
Yes, I can hear you.
Okay, dear attendees, you can write your questions on the chat box on the right-hand side of your screens. And if you're watching this video on YouTube, we are going to share these questions in the description box below, so you can check the questions from there. I would like to thank Dr. Kılınç for this nice talk, because Siberia, Yakutia, these regions are quite important, especially for the Eurasian to America crossroad is quite important for population genetics. So I think it was interesting.
Thank you very much.
Actually, I had one general question, but you kind of answered it during your talk, but I'm gonna ask it again anyway. So at the beginning of your presentation, you mentioned about the contaminations that can be encountered during human sequence isolation. I think it was on page 6. So is it so common that to find an ancient bacteria on a human sample, for example? Is it something that regularly happens, or is it unheard of?
Normally, in human population studies that we do with ancient human genomes, we do not examine this. Actually, I need to be honest, because we don't analyze them, because our questions are related to human population dynamics. So if there are some sequencing reads that are contaminating and that are coming from an ancient bacteria, they will not interfere with our results, since we will be dealing with the human SNPs. But there are some recent studies that people are working on ancient microbes. But it is a very recent field, so people are discovering new things. But I don't know. So what did they find so far? So it might be, of course. But for that, you need to do some kind of like analysis that you generate like trees to find where the microbe that you're interested in is located on the tree that you're examining. So you need to do such kind of things. But in our questions, since we didn't interested in that, I don't know if our samples had this or not.
Okay, thank you so much.
Okay.
If you're asking question, please use this little icon, hand icon down on the chat box, so that we will know that somebody is going to ask a question.
Was it too complicated or too easy? Nobody asked.
It was regular, I guess. Now there is a question from Yasemin. Can you see the chat box right now, or should I read it for you?
You can read, or I can just stop sharing and maybe. Thank you, Yasemin. Contamination. I wonder if it is needed specific equipment to do it. So, do you mean like equipment, like a software that we do, or like a machine or something to check if there is contamination? So if you mean that if there is a machine or something, there isn't. So what we do is first we isolate the DNA in the clean lab. We try to be sure about there isn't any contamination. So we do whatever we can, like wearing gloves and then decontaminating all the surfaces using a clean lab and excavating the sample under clean conditions. So these are the precautions that we can follow. But even under those precautions, there can still some kind of contamination at some degree. But this is not a case in recent years. It was a big problem at the beginnings of the ancient DNA studies, but now it is not a big problem. So there isn't any kind of machine. You can just isolate the DNA, and then you can run it on like bioanalyzer to see the fragments. And after sequencing, what we do is using some bioinformatics approaches to analyze the distribution of the read lengths or to analyze the ancient DNA specific mutation patterns or some kind of like mitochondrial DNA based or nuclear DNA based analysis to see that reads, sequencing reads that we produced are really coming from an authentic DNA. So this is just the thing that we can do, but there isn't any machine that you can separate the endogenous and like the normal, not normal, but endogenous and like the modern DNA sequences. No machine. Thank you too.
Okay. I guess we don't have any other question right now.
Okay.
So again, I would like to thank you, Dr. Kılınç, for taking that time and making this webinar. It was quite important for us, and I guess we can stop here. Oh, we have another question.
Yes, Munta. We know each other. Thank you for joining, first of all. The question is, you explained everything clearly. You talked about big data set, about thousands of individuals. So my question is about how the samples are prepared. Have you been involved in sample preparation and DNA isolation part? Which parts are preferred? Bones, hairs? So many questions. So the question is about how the samples are prepared. I'm not involved in the sample preparation and DNA isolation part, and I'm mostly the one who is running the bioinformatics analysis and population genetics analysis and writing the story. But the DNA like isolation part and the sample preparation part are done by other friends, colleagues. But for that, what they do is taking the samples to the lab and then cleaning the samples. And what I know is that they are getting some samples from the tooth or like long bones, whatever soft tissue that you have in your hand. And for DNA isolation, normal kits, like for normal DNA isolation kits are used, but with some kind of modifications that can be specific to ancient DNA to increase the yield of the DNA. Bones or hairs, so whatever you have in your hand, but mostly the temporal bone is preferred, and a bone inside the ear is also preferred. Petrous bone is a very good source of ancient DNA, but if there isn't any petrous, you can go for the molars and like teeth, and also you can extract the DNA from femurs or like tibia, ulna, whatever kind long bone tissue. So, but if you have hair, you can also try to obtain ancient DNA from that one, and you can do that. So, any kind of soft tissue, but the petrous and the molars are preferred first. On the so consider everything and depend on competence. I didn't understand that part. You consider everything. Yes, we consider everything. So if which sample we have, we just isolate the DNA. Thank you very much for this question too. Hope it was clear.
I think I have one more question.
Yeah, sure.
I was wondering that how genetically similar the current inhabitants of the Lake Baikal area or Siberia and the, for example, Kyrgyzstan person.
So
Is it something that you...
Can you repeat the question? Sorry, I lost your voice.
How similar genetically this Kyrgyzstan person and the current inhabitants of Siberia or Lake Baikal area? Is it something that you checked?
Yes, sure. I can maybe share my screen again. So this Kyrgyzstan individual. So here we have some like Siberian populations when we go back to PCA. Here on this PCA, what we have is that the small points are, each of the small points is representing one individual from a modern population. So for example, here we have some East Asian populations. So I didn't put the names of all populations because it can be complicated. There are too many different populations. And even if you work with the ancient DNA, you always mix up with the population names and everything. But because of that, I didn't want to be too complicated. But in general, here each dot is representing one individual. And this group is, for example, East Asian populations. Here we have some South Asian populations. And here we have the West Eurasian populations on this part of the figure. And here we have some America populations. In F3 statistics, this is a kind of statistics that you like imagine a tree and then you measure the sharing of alleles between different groups. So this YRI is representing a Yoruba individual from Africa, and this is an out group. So after separated from out group, here we test if Kyrgyzstan is sharing alleles with which population. And for POP-X, we put different modern populations here. In the PCA, we don't see any kind of affinity around because there is just one population. But this one was, so here on the PCA, it is like the Kyrgyzstan is sitting on a separate place. But in this F3 statistics, what we see is that this Kyrgyzstan individual is sharing more alleles, more drift with the Native American population. So it was more genetically similar to the Native American populations. And also it was interesting that this is similar to share more alleles with the Native Americans. And also, since we know that this Colima individual here is the ancestor of the Native Americans. And if you remember, we modeled this Colima individual as a mixture of Kyrgyzstan and the East Asia. So it seems like this Kyrgyzstan genetically contributed to the ancestors of Native Americans and shared alleles like more similar with the Native American modern populations compared to the Asia populations. Was that clear now?
Yeah, it's clear. Thanks so much.
Thank you very much.
There is another question from Sheval.
Thank you very much, Sheval, for joining. And we are also friends, I guess. So this was about, I wonder whether we know that those individuals are related or not. So we checked the relatedness. There were two related in the whole data set. We examined a data set of 40 individuals. Probably there were 2 or 3 related individuals. In Yakutia, most of the individuals were related. So we had also some mother-father-child trios, but in the population genetics analysis, we removed the related individuals from the analysis. In the supplementary table of the paper, probably in the first or second supplementary table, we wrote about it, which individuals were related to each other. I don't remember exactly the name of the individuals, but there were relatives in the data set.
Okay. I think we are done for today. I would like to again thank you on behalf of RSG-Türkiye for joining us and making this webinar. And I would like to thank our attendees as well for their interest. And for today, we can stop right now.
Okay, thank you very much again for your invitation and for everyone for joining today. Thank you.
Thank you. See you in another video or another webinar.
Yeah. Egenas is raising hand, but maybe.
Yeah, there is one more question. Okay.
Why do we remove relatives from data sets? So in the population genetics analysis, what we do, so most of the population genetics analysis that we run, like F statistics, for example, they are based on the allele frequencies. So if you include the relatives in those analyses, it is like using the repeated samples, like the same individuals. So we just remove the relatives, but if there are two individuals and if two of them are related to each other, we keep just one of the individuals to represent a more uniform data set. Because if you, for example, compare two different groups, in one group you have 5 relatives, 5 individuals, and all of them are related to each other. And then in the other group you have 5 individuals, but all of them are like different individuals, they are not related. Then this will be like the related carrying in the population will be like a homogeneous group of just relatives. So just to prevent over-representation of a single population, we remove them for the frequency-based statistics. Thank you very much again.
Is there anyone else who are writing question? You can use the little hand icon down on the chat box.
Thank you.
I think this time we really don't have any other question.
Okay,
I just wanted to be sure everybody, please, if you are writing a question, please use the little hand icon down in the chat box so that we will know somebody is asking a question.
I guess nobody is asking. Yeah. Thank you very much.
Thank you so much again for making this webinar. We would like to see you again, maybe next year with new results.
Yes, thank you very much. I would love to do that.
Okay, thanks so much for joining, dear our attendees, and I guess we can stop here right now.
Thank you. Bye.
Thank you.